So, what if I told you that a diet celebrated for starving cancer in one part of your body acts like, well, pure rocket fuel for tumors just a few inches away?
I mean, it completely upends how we usually think about nutrition, right? We desperately want diets to be binary, like healthy or unhealthy.
Right, good or bad for you. We want this simple software update. You just download the cut back on sugar patch or, the eat more plants update, and you just assume the whole system runs better.
Yeah, exactly. But when you step into the world of targeted cellular metabolism and genetic predisposition, that simple update can actually start causing massive glitches in very specific hardware.
Because, diets don't just provide passive calories
Right,
here.
not at all. The foods we eat are these complex chemical signals. They actively reprogram our cellular machinery right at the genetic level.
And that actually brings us to the mission of today's deep dive. We are dissecting a really, really dense, brand new study from Nature. This was published in July 2026.
It's a fascinating paper.
It really is. It's titled Ketogenic Diet Mediates Intestinal Tumorigenesis Through Lipids, Not Ketones. Now, the ketogenic diet, keto, it's everywhere.
Absolutely everywhere.
Yeah, it's praised for weight loss, managing certain neurological conditions, and even, in some circles, for cancer prevention. But the source material we have today reveals this terrifying paradox.
That's a massive shift.
Okay, let's unpack this. In the small intestine, a strict keto diet actually accelerates tumor growth in genetically susceptible individuals.
Which is just a profound shift in our understanding.
Right.
So today we're going to explore exactly how a diet composed of 80% lard rewires the gut, step by step, molecule by molecule.
Right.
We're going to dismantle the entire metabolic engine to see what is really driving this explosion of tumors.
To do that, we kind of need to set the stage. I want you, the listener, to visualize your small intestine. Don't think of it as just a digestive tube.
Right, that's too simple.
Yeah, think of the intestinal lining as a highly dynamic microscopic factory. And this factory is so intensely active that the entire lining completely replaces itself every, 4 to 5 days?
Yeah, every 4 to 5 days. It is an absolute masterpiece of biological engineering.
It's insane. But a factory that active, it needs a massive specialized workforce.
Exactly. And in the gut lining, the most important workers are the intestinal stem cells. Specifically, they're called the LGR5 plus stem cells.
Okay, LGR5 plus, yeah.
Right. If you were to look at the lining of the gut under a microscope, you'd see these deep microscopic valleys called crypts. That is where these stem cells live. And normally their regenerative power is tightly, meticulously controlled.
They just hum along.
Yeah, they divide, they migrate up the walls of the valley, and they mature into the specialized cells that, actually absorb your food.
But like any factory, if the management software gets corrupted, things go really wrong. And that brings us to a very specific genetic defect known as FAP.
Right, familial adenomatous polyposis.
Yeah, FAP. It's caused by a mutation in a tumor suppressor gene called the APIC gene, APC. And I like to think of this AP gene as like the emergency brake on those stem cells.
That's a highly accurate way to picture it, actually. The apigene normally helps degrade proteins that tell the cell to divide.
It keeps them in check.
Exactly. So when that apic brake is lost due to a mutation, those stem cells go rogue. They act as if someone put a brick on the gas pedal. They just multiply uncontrollably and form adenomas, which are basically early-stage tumors.
And what's fascinating here is the clinical reality for these patients. Because this mutation affects the entire digestive tract, FAP patients are almost guaranteed to get colon cancer.
Yeah, it's a very difficult diagnosis.
So to save their lives, surgeons will often remove their colon entirely. But tragically, even without a colon, they often still develop and ultimately pass away from tumors in their small intestine.
Which is why finding ways to protect the small intestine is the absolute top priority in this field.
But wait, this raises a massive question for me. I was pushing back hard on this when I was reading through the paper. We know that fasting and calorie restriction usually help cellular repair, right?
Generally speaking, yes. They suppress tumor growth.
And the ketogenic diet famously mimics the metabolic state of fasting. It lowers insulin, it shifts the body's fuel source. So shouldn't a keto diet, by lowering those growth signals, actually protect these mutant stem cells from turning cancerous?
You aren't alone in that thought. I mean, that is exactly the logical assumption the scientific community held for a long time.
It just makes intuitive sense.
It does. Because the keto diet dramatically lowers circulating insulin, and insulin is a hormone that promotes cellular growth, you would naturally assume it slows down cell division. But, well, Biology loves to defy our simple assumptions.
Okay, before we dive into how that assumption gets completely shattered, I need to pause and make sure I have this foundational mechanism straight. For you listening, let's do a quick recap. Okay, so bullet point 1. We've got a gut lining that is constantly rebuilding itself using stem cells down in these little microscopic valleys.
The crypts.
Right, the crypts. Bullet point 2. When there's a mutation in the APAC gene, the breaks are cut and those stem cells form dangerous tumors, especially lethal in the small intestine for people with FAP.
Spot on.
And the final point. The big question we're looking at is what happens when we flood those vulnerable, breakless stem cells with a ketogenic diet?
Which leads directly into the researchers' first major experiment.
Yeah.
They took mice that were genetically engineered to have this exact APAC mutation, so simulating the human FAP condition.
Okay.
They fed one group a normal, purified control diet, and they fed the other group a strict ketogenic diet. And in this specific animal model, that keto diet was formulated as 80% lard.
Wait, hold on. 80% lard.
Yeah, no carbs, moderate protein, and massive amounts of fat.
That sounds extreme. Is that even a realistic comparison to a human keto diet? I mean, nobody is just eating spoonfuls of pure lard.
It's a great point, and it's a really common critique of animal studies. But in this context, the researchers aren't trying to replicate a human recipe. They're trying to isolate a biological variable.
Oh, I see.
Right. If you give mice a human keto diet full of nuts, avocados, cheese, you introduce thousands of confounding variables, different fibers, plant toxins, dairy proteins.
So you wouldn't know what's actually causing the effect.
Exactly. By using a highly purified lard diet, they can say with absolute certainty that any changes they observe are strictly due to the macronutrient shift, the extreme lack of carbs, and the extreme presence of dietary fat.
Got it. Okay, so they isolate the fat variable, feed these APAC mutant mice the lard, and the result was drastic. I mean, the charts in the paper are actually quite staggering.
They really are.
The mice on the keto diet didn't just get a few more tumors. They suffered a massive explosion of small intestine tumors, like a complete acceleration. Their survival rates plummeted compared to the mice on the normal controlled diet.
It was incredibly clear-cut.
The keto diet wasn't protecting them at all. It was actively killing them faster.
And to figure out exactly why, the researchers had to play metabolic detective. When a biological crime like this occurs, you first look at the most obvious suspect.
Right.
In this case, the defining feature of any ketogenic diet, the ketones themselves. When you stop eating carbs, your body's glycogen stores deplete. Your liver then starts breaking down fat into alternative fuel molecules called ketone bodies.
And the two main ones are, BHB and AHAC?
Yep. BHB, or beta-hydroxybutyrate, and AHAC, or acetoacetate. Those are the ones circulating in your blood.
And here's where it gets really interesting, because previous studies have actually pointed to BHB as the good guy.
Right, the tumor suppressor.
Yeah, there was this prevailing idea in the literature that BHB binds to a specific receptor on the surface of cells called HCAR2, and that binding action actually suppresses colon cancer.
It's a known mechanism.
So the researchers initially hypothesized maybe the ketones are trying to help, but something else in the diet is overriding them.
Right, but to test that hypothesis, they couldn't just guess. They needed to completely dismantle the body's ketone production machinery.
How do they do that?
We have to look at the physical process of ketogenesis. Imagine a highly specific, complex machine on the biological factory floor whose only job is to turn raw fat materials into ketones.
Okay, I'm picturing it.
That machine is an enzyme called HMGCS2. It is the critical, rate-limiting step. Without that specific enzyme, it is biologically impossible to make ketones from fat.
So how do you turn off a microscopic machine inside a living animal?
The researchers use something called a Krelok system.
Krelok.
Yeah, you can think of it as incredibly precise genetic scissors. They use these scissors to knock out or delete the HMGCS2 enzyme. First, they deleted it just in the intestine, which they call the IKO model.
Small I for intestine, KO for knockout.
Exactly. Then they deleted it systemically, meaning in the liver as well, which they call the DKO or double knockout model. They essentially went in and smashed the ketone factories. Zero ketone production, no matter how much fat the mice ate.
That's wild. But they didn't stop there, did they?
No, they were incredibly thorough. They thought, what if the cells are still somehow finding and using ambient ketones from somewhere else? So they went after the machinery for ketolysis as well.
Ketolysis, that's the process of how cells actually eat or burn ketones for fuel, right?
Yes. They used their genetic scissors again to knock out the specific enzymes required for this, known as BDH1 and OXCT1.
BDH1 and OXCT1. Okay.
If you think of ketones as a specialized type of gasoline, knocking out those enzymes is like completely removing the engine that burns that gasoline.
Okay, so paint the picture for me. They now have these mutant mice eating a diet that is 80% lard. But the mice literally cannot make ketones, and even if they somehow could, their cells lack the engines to burn them.
Right.
What happened to the tumors?
The tumor explosion still happened. It was identical.
Wow.
Yeah, whether the mice couldn't make ketones or couldn't use ketones, the keto diet still massively accelerated the cancer. The researchers even did the reverse experiment, just to be absolutely sure.
Well, what was that?
They genetically forced normal mice to overproduce ketones while eating a regular low-fat diet.
Did those extra circulating ketones cause tumors?
No, completely benign.
Okay, but what about that receptor you mentioned earlier, the HCAR2 receptor that supposedly uses ketones to suppress cancer?
They looked for it. They used advanced single-cell RNA sequencing to map out the small intestine. And that receptor is practically non-existent there.
Wait, really?
Yeah, it's not on the stem cells. It's a ghost in this specific tissue.
Wow. Okay, let me pause again and synthesize this. Maybe do our second quick recap for you listening, because this is a massive twist.
It really is.
So, 1, we put these genetically vulnerable mice on a keto diet, and their small intestine tumors explode.
Yep.
2, the researchers meticulously went in with genetic scissors and deleted the ketone factories, the HMGCS2, and then deleted the ketone engines, the BDH1 and OXCT1.
Right, dismantled it all.
And 3, the conclusion. The tumors still grew just as fast. So ketones are entirely innocent bystanders. They're just hanging around at the scene of the crime because they get produced when you eat fat, but they are not the ones pulling the trigger.
Which forces us to look at the only other major suspect left in the room.
Right. If it isn't the lack of carbs causing the tumors, and it isn't the presence of ketones, it has to be the fat itself. The diet is 80% lard.
Exactly.
But here's where I get confused. Fat is just a blob of stored energy, right? How does a blob of fat hack into a stem cell's operating system and tell it to mutate?
That is a very common misconception. Fat is not just passive inert energy. Dietary lipids are highly active signaling molecules.
Signaling molecules, okay.
And this paper introduces us to the cellular switchboard that receives this fat signal, the PPAR system.
PPAR.
Yeah, these are transcription factors, specifically PPAR alpha, delta, and gamma.
A transcription factor, meaning it reads the environment and then alters how the DNA is expressed.
Exactly. Think of PPAR as an aggressive cellular middle manager.
I love this analogy.
Under normal, balanced dietary conditions, this manager just hums along quietly in the background, keeping things orderly. But when the gut is suddenly flooded with a massive, continuous influx of dietary lipids from a keto diet,
The switchboard lights up?
the switchboard lights up. The lipids literally bind to the PPAR manager. The manager then travels directly into the nucleus of the stem cell, grabs the DNA, and starts shouting orders.
We have a massive surplus of fat. We have unlimited resources. Start building, start expanding, multiply right now.
Exactly. It pushes the stem cells into absolute overdrive. And the researchers didn't just theorize this, they proved it. They created what they called a triple knockout mouse, the PPAR3KO.
So they deleted all 3.
Yes, they went in with their genetic scissors and deleted all 3 of those PPAR managers in the intestine. And when they did that, the stem cells lost their hyperactive keto superpowers. They stopped expanding uncontrollably.
That's incredible.
They also proved it pharmacologically, which is vital for potential human treatments. They took mice currently on the keto diet and gave them a drug called GSK3787.
GSK3787, what does that do?
It's a PPAR antagonist. It essentially puts a gag on that middle manager.
Nice.
And successfully, when the manager was gagged, the tumor shrank. They even did the inverse. They gave mice on a normal, low-fat diet a drug that artificially activates PPAR, and those mice grew larger tumors.
Okay, so what does this all mean? We've established a chain of command. The dietary fat enters the gut. It activates the PPAR manager, and the manager yells at the cell's DNA to grow tumors.
Right.
But how does PPAR actually build those tumors? What is a physical mechanism on the factory floor that turns a genetic command into a physical mass of cancer?
This brings us to the final and arguably most crucial piece of the mechanistic puzzle. Fatty acid oxidation, or FAO. This is the biological process by which cells break down long-chain fatty acids to produce immense amounts of cellular energy. PPAR is the manager giving the orders, but fatty acid oxidation is the engine room generating the power to carry those orders out.
Okay, I think I need a visual analogy for this, because to burn fat for energy, the fat molecules have to enter the cell's internal furnace, which we know as the mitochondria.
Right, the powerhouse of the cell.
Exactly. But long-chain fats can't just casually stroll through the mitochondrial membrane. The furnace has a very strict bouncer working the dual.
I like where this is going.
And that bouncer is an enzyme named CPT1A. If the fat doesn't get a specific tag from the CPT1A bouncer, it doesn't get into the furnace and it doesn't get burned.
That is a brilliant way to visualize it. CPT1A is the rate-limiting transport enzyme. It attaches a carnitine tag to the fat so it can cross the double membrane of the mitochondria.
Okay, so the tag is like the VIP wristband.
Exactly. Now, in the stem cells of the keto-fed mice, the researchers found that those signals from the PPAR manager caused a massive upregulation of CPT1A.
So the cells were essentially building more doors and hiring an army of bouncers to shove as much fat into the furnace as physically possible.
Precisely. So the researchers set up their ultimate proof. They engineered a mouse that specifically lacked this bouncer in its intestines. The CPT1A IKO mouse.
No bouncer.
Right. When they fed these modified mice the extreme 80% lard keto diet, the fat still arrived in the gut. The researchers checked the cellular lipidomics, the fats like triglycerides, and they were still physically accumulating inside the cells.
So the fat arrived at the club, but because the CPT1A bouncer was missing, it couldn't get into the furnace?
And the result was definitive. Without CPT1A, without the ability to transport the fat into the furnace, the keto diet could no longer cause the tumor explosion.
Oh, wow.
Yeah, the mice lived significantly longer, and the massive tumor burden basically vanished. It returned to the baseline levels of the control diet.
That is a staggering finding, because it proves that the tumors aren't caused by the cells simply storing fat or getting clogged up by it. The cancer is driven by the specific active process of burning that fat via fatty acid oxidation.
Exactly. Unregulated cell division, which is what cancer is, requires a colossal amount of energy.
Yeah.
The intense metabolic output of burning long-chain fats provides that energy.
Right.
But it goes deeper than just ATP. When you hyperactivate fatty acid oxidation, you flood the cell with byproducts like acetyl-CoA.
Acetyl-CoA, right.
And these byproducts can actually alter the epigenetics of the stem cell, changing how its DNA is spooled and read, fundamentally locking it into a cancerous trajectory.
Yeah.
The requirement is absolute. You must actively burn the fat to grow this specific tumor.
Okay, let me make sure I have this whole chain of events locked in my head. This is our final mini recap for you listening.
Let's hear it.
First, the massive amount of dietary fat from the keto diet enters the cell and activates the PPAR switchboard. Second, that PPAR manager screams at the cell to ramp up the engine room, specifically demanding more fatty acid oxidation.
Perfect.
And finally, the enzyme CPT1A acts as the gatekeeper to the furnace. When the researchers blocked CPT1A, the fat couldn't burn and the tumor stopped growing. This proves definitively that the active burning fat is the true fuel for this specific cancer, entirely independent of ketones.
You've got it perfectly, which actually brings up a glaring, massive biological contradiction that we absolutely have to address.
Yes, because earlier you mentioned that some studies show a keto diet actually suppresses tumors in the colon.
Right.
How is it biologically possible for the exact same diet, the exact same macronutrients entering the same digestive tract to act as a tumor suppressor in the colon, but act as a massive tumor promoter just a few inches away in the small intestine?
It all comes down to tissue-specific biological context. We often lump the digestive tract together, but the small intestine and the colon are two vastly different organs.
Yeah.
They have entirely different mandates.
How so?
Well, the small intestine is built to aggressively absorb nutrients. It is primed, evolutionarily, to snatch up lipids and burn them for energy to drive its massive, rapid cellular turnover.
And the colon?
The colon is primarily designed for fluid balance, electrolyte recovery, and managing your microbiome. It doesn't have the same mandate to aggressively absorb and burn raw fat for fuel.
So they react completely differently to the exact same environment.
Exactly. When the colon is exposed to a keto diet or even just a standard high-fat diet, the tumor burden actually goes down. And crucially, the researchers in this paper proved that this suppressive effect in the colon is also independent of ketones.
Oh, really?
Yeah. When they looked at the cellular lipid profile in the colon on a keto diet, it barely changed compared to the massive fat accumulation seen in the small intestine. The small intestine greedily absorbs and burns the fat, which drives the cancer. The colon largely ignores the fat, resulting in a completely different metabolic outcome.
Wow. If we pull back and look at the bigger clinical picture here, the implications are just chilling. Let's go back to those human patients we talked about at the beginning, people with FAP.
Right, the genetic mutation.
A doctor performs a preventative colectomy. They remove the colon to save the patient from inevitable colon cancer, so they no longer have a colon, but their small intestine remains intact.
Right. And imagine that patient, desperately looking for ways to stay healthy, reads a headline on a blog that says, "Keto starves cancer."
Oh, wow. So they adopt a strict ketogenic diet, thinking it's a universally anti-tumor software update for their body.
And by doing so, they might be inadvertently throwing gasoline on the exact organ where they are now most vulnerable. They are force-feeding the exact fatty acid oxidation pathway that drives small intestine adenomas.
It highlights beautifully and terrifyingly why we have to move beyond the idea of good or bad diets. Nutrition is not a blunt instrument. It is a highly specific chemical code.
Absolutely.
You cannot reduce the biological consequences of a complex diet down to the effects of a single molecule, like a ketone.
It is entirely about context. A dietary intervention that wonderfully promotes tissue regeneration, say, by boosting stem cell activity, might simultaneously reshape the metabolic landscape to favor tumor initiation if you happen to have the wrong genetic mutation.
Right, because accelerated repair means accelerated cell division.
Exactly. And if your FCP brakes are cut, accelerated division means accelerated cancer.
As we wrap up this deep dive, I want to leave you, the listener, with a question to really chew on. We started this conversation by talking about our expectation of precision, our deep desire for diets to be universally good software updates.
If we connect this to the bigger picture, we've just seen that the exact same nutritional code can fix one organ and completely crash another.
Exactly. So here's my question for you to mull over. If a single diet can simultaneously starve tumors in your colon but actively feed tumors in your small intestine, how does that change the way you view the one-size-fits-all dietary advice we see pushed every single day on social media? Because when it comes to biology, the simple answers are usually wrong and the muddy waters are where the real truth hides.